Hi, everybody, Dr. Harmon. So, this is our last set of notes for Unit 1, and we will be talking about nucleic acids. So, let me get my screen shared, and we will get started. Okay, so here we're talking about nucleic acids, which is out of our topic guide. This would be topic 6. Nucleic acids are polymers made of nucleotide monomers. Okay. So we're going to build out of a monomer called a nucleotide. Same concept, uses the same process, so we're still using the dehydration reaction to build them, hydrolysis to break them apart. It's just we've changed the monomer. Okay. The function of our nucleic acids is to store, transmit, and express hereditary information. So this is all about our genes, all about that genetic information that tells us what we are and how we're made and all of that. So we have two key forms of nucleic acids. Our deoxyribonucleic acid, known as DNA, and our ribonucleic acid, known as RNA. Okay. So in terms of components of nucleic acids, we have our nucleotides, which become our polynucleotides, which become our nucleic acids. So we've got steps just like we did in all the other ones, where we start with our monomers and we can put a few together to make a multi-polymer. Then ultimately we put so many together that it becomes the nucleic acid. Okay. So a nucleotide has 3 key parts. We have our nitrogenous base. There is a 5-carbon sugar known as a pentose sugar. And we have a phosphate group, or a series of phosphate groups, depending on what we're building with the nucleotides. We found that in polynucleotides, each monomer has only 1 phosphate group, but we have another molecule that's critical to life that we build out of nucleotide that has more than 1 phosphate group. Okay. So first we want to look at the nitrogenous base. And so we have a variety. Here you can see our cytosine, thymine, uracil, adenine, and guanine. Some of these may sound familiar from when you took your original biology class. Some of them you may not remember their actual names, but if I were to say A, T, C, G, and U, then you'd go, oh yeah, I remember us doing something like that. And some of you are probably still going, we talked about that? We did. Okay, so we have 2 key types of nitrogenous bases. We have what we call the pyrimidines and we have the purines. Okay. Pyrimidines will have 1 ring with 6 atoms. So cytosine, thymine, and uracil, as you can see, all each have a single ring of 6 atoms. So they are all going to be our pyrimidines. Cytosine, thymine, and uracil. Thymine is only found in DNA. Uracil is only found in RNA. Thymine's kind of like the elephant in the room. If we put an elephant in our classroom, we couldn't fit it through the door to get it out. So thymine is shaped such that it cannot leave the nucleus. Uracil basically does the same thing, but it's a little bit smaller. And if you look at the structures, you can see how that works. And so it can leave the nucleus. That's why we use it for RNA. Then our other type of nitrogenous base would be our purines. Purines have 1 ring with 6 atoms bonded to 1 ring with 5 atoms. So you'll notice Here we have 6 and 5. And so that would be our adenine and our guanine. So cytosine, adenine, and guanine are found in both DNA and RNA. Thymine is only in DNA. Uracil is only in RNA. Okay. And then, remember, the next part of our nitrogenous base was the 5-carbon sugar. This is what one of the key factors that determines whether it's RNA or DNA. A sugar is going to be bonded to the base. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. Look at the two molecules. One of them has one more oxygen than the other. So we have ribose, and then deoxy. When we say deoxy, that means we took away an oxygen. So then we have our deoxyribose sugar. This is where those names of ribonucleic acid and deoxyribonucleic acid come from, is based on the sugars. So notice deoxyribose has a slightly different structure, and one of the key differences in the structure of the sugar is that oxygen is missing. Ribose has an oxygen, deoxyribose doesn't. And amazingly enough, that oxygen really does play a big role in terms of the structure and function of these molecules. Okay. And then the third part of our nucleotide was the phosphate group. The phosphate group is added to the 5' carbon of the sugar. This is the one that's attached to the base to form a nucleotide. So you'll notice we attach off of each of these points on this ring is a sugar. Remember, we talked about that before, that when we have these bends or twists, that's a carbon at each point. So carbons, carbons, carbons. And this is considered our 5' carbon that the phosphate group will be attached to. And notice the nitrogenous base is attached on the opposite carbon on that sugar. Okay. So we have what we call the nucleoside portion, and then we have the phosphate group. So the nucleoside is the part without the phosphate group on it. So if we start adding these individual nucleotides together, we make a polynucleotide. So phosphate groups will link to the adjacent nucleotide. So notice that basically we have a chain of, here's our sugar that has the phosphate group hanging off. That phosphate group will also then connect to the next sugar. It has its phosphate group hanging off, and that phosphate group connects to the next sugar. We call these phosphodiester linkages. They provide directionality. So whenever we talk about our nucleic acids, we often use markers. We call it like 5' and 3'. Those are the key, and they're based on which sugar, which carbon in the sugar we're basing off of. And so this provides a directionality of the molecule. And we always go 5' to 3' when we talk about it. So you'll see our sugar phosphate backbone, and you see our nitrogenous bases hang off the other side. The 5' is going to be the phosphate end because it's attached to the 5' 5-carbon of the sugar. And the 3' is what we call the hydroxyl end because there's going to be a hydroxyl group right there. Okay, the sequence of the bases along the DNA or the messenger RNA is unique for each gene. So we're looking at which nitrogenous base is put in there. Is it an A, T, C, C, G, whatever that sequence is, it's going to be unique for the gene that it is coding. It will then dictate that amino acid sequence. So it's going to tell what order we put those amino acids in to make our protein. So it dictates the primary structure of the protein. And therefore, because the primary structure is what really determines the overall three-dimensional shape of the protein, that sequence of bases in the nucleic acid is going to dictate or determine the 3D structure of the protein. Okay. So looking specifically at DNA, we have 2 polynucleotides. And these 2 will kind of face each other and they form what we call a double helix. Sometimes you'll hear it termed as a twisted ladder. That's a more common descriptive term, but the official terminology is double helix. The strands are anti-parallel. If you'll look at this bottom right picture, we go from 3' up to 5' on one side. On the other side, 3' at the top and 5' at the bottom. They are going in opposite directions, hence anti-parallel. These 2 nitrogenous base sides, the 2 strands, are actually held together by hydrogen bonds. And remember, when we talked about the hydrogen bonds, one of the characteristics we pointed out is that they were weaker than our covalent and our ionic bonds. And the idea is that they can be broken and reformed much easier. And so we can open and close the 2 strands of the DNA, the double helix, more easily because of the hydrogen bonds that hold these molecules together. As a rule, and you hopefully remember this, but you might not, cytosine binds to guanine, adenine binds to thymine. ATCG, apple tree chewing gum, right? Then with RNA, RNA is a single-stranded polynucleotide, so we only have 1 strand of nucleotides here. They're going to be much more variable in shape because we're not having to interact the 2 molecules. And that variation is due to base pairing within the RNA. So a cytosine within the RNA can attract to a guanine in that same RNA molecule. So they can actually connect with each other while they're not being read or used. Adenine will bond to uracil in RNA. Remember, thymine is in DNA. Uracil takes the exact same spot in RNA. So adenine bonds with uracil and cytosine will bond with guanine just like before. So here we have our chewing gum, but we have our apples wearing underwear. Okay, so here's practice. You're given a segment of DNA, 5', C, A, T, G, T, C, A, C, 3'. Okay. What would be the complementary strand? So what would pair? So think about it. We go from 5' to 3'. C is going to pair with, A is going to pair with, T is going to pair with. You get the concept. So that's how you determine the complementary strand. Okay. So we have 3' to 5' because remember it's always going to be anti-parallel, opposite, so the numbers reverse. It's going to be G, T, A, C, A, G, T, T, G. It's going to pair with. Whenever we ask for complementary, we're always talking about what pairs with what pairs with. Okay, so we've gone through all of the different macromolecules, so it's time to kind of put it all together and look at how they relate to each other. So we're going to look at macromolecule, what elements are involved, what the monomer is, and what the key polymers are. Okay, so for carbohydrates, we have carbon, hydrogen, and oxygen, CHO. The monomer or subunit is a monosaccharide, one sugar monosaccharide. And our key polymer are polysaccharides. And we have a variety of different polysaccharides we talked about. Proteins use carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur for those bonds. But we typically refer to this one as CHON, carbon, hydrogen, oxygen, and nitrogen. We don't typically include the sulfur when we just do our little abbreviation. The monomer is the amino acids. The polymer would be polypeptides. Lipids, and remember lipids we said aren't exactly a perfect monomer polymer concept, but so the elements are carbon, hydrogen, and oxygen. Those are the key, so it's another CHO, but some of the lipids like phospholipids will also use phosphorus. So we do see some other elements pop in, but our key ones are carbon, hydrogen, oxygen, CHO. And especially when we're talking about the triglycerides or the base structure for phospholipids, we have the glycerol, that's the alcohol with fatty acid chains, fatty acids. The polymer, it doesn't really contain any true polymers. We're going to make different types of lipids based on how we combine these different molecules. And then we have our nucleic acids. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. That's CHUMP. Okay. The monomer is the nucleotides. And the polymer would be DNA or RNA. Okay. So that kind of gives you the big picture for nucleic acids and finishes up all of our key aspects that we wanted to focus on for our notes in Unit 1.